Have you ever walked through, say, a cornfield and noticed something just off? Like a cob that looks like it's covered in this weird, black, dusty, almost soot-like stuff.
Yeah, it's pretty striking when you see it.
It is unsettling, right? It looks like something's gone really wrong. But what if I told you that that very thing is actually this complex, fascinating organism, one that can cause millions in damage, but also end up as a gourmet delicacy? And even be like a superstar for scientific research. Okay, let's unpack this.
Well, what's truly fascinating here is exactly that duality, how something that looks so destructive and is destructive in many ways can also hold such surprising value, even, yeah, delicious value sometimes.
Right.
These fungi are definitely infamous plant pathogens, no doubt, but they're also just invaluable tools for geneticists, for molecular biologists. It's this remarkable hidden world, really, beneath what most people would just, call mold or blight.
Exactly. So our mission today is to do a deep dive into this hidden world, the world of smut fungi, specifically from the phylum Basidiomycota.
We've got some really rich source material here, and we want to help you understand what these mysterious organisms are, how they actually work, and why they matter so much, both economically for our food,
Critically important there.
and scientifically for just understanding life itself. So think of this as a journey. We'll start big picture, their impact, then drill down to the microscopic details, and we'll keep connecting it back to why you should care, why this knowledge matters. Okay, so let's start with the name, smut. It sounds pretty unpleasant.
It does.
And it comes right from those black, dusty masses of spores. They're called teliospores, which really do look like soot on the diseased plants. That's the classic image. But don't let that simple name fool you. These are biotrophic pathogens. Okay, that sounds a bit technical. But think of it like this. They're kind of like biological vampires. They need to feed on living host plants to really thrive. That's a good way to put it. They draw nutrients right from the living plant, often keeping it alive for as long as possible.
But it's not always about the living host, is it?
No, that's a key point. Smuts also have saprobic phases. So while, yeah, their main gig is feeding on living plants, they can also switch gears and act like decomposers, growing on dead organic stuff if the conditions are right.
Huh. Flexible.
Very. And their reach is just astonishing. We're talking something like 1,200 known species spread across more than 50 different groups or genera. And collectively, they attack around 4,000 species of flowering plants.
4,000.
Yeah, across over 75 plant families. They are basically everywhere plants are. Truly ubiquitous.
And I guess when something is that widespread, the economic consequences must be pretty significant.
Oh, absolutely. The damage adds up incredibly fast. These smuts cause literally millions upon millions of dollars worth of damage to essential food crops and even ornamental plants every single year.
Wow.
Take corn smut, for instance. That's caused by Ustalagomades. You find it wherever corn grows, basically. Big farms, small gardens, doesn't matter. And it can cause serious losses, especially for sweet corn, where it forms those pretty unsightly tumor-like growths called galls on the ears.
Yeah, I can picture those sort of lumpy, grayish black masses.
Exactly that. But maybe even more impactful on a global scale are the bunt diseases of wheat. These are caused by various Tillacea species. And honestly, they're as big a problem for wheat production worldwide as the infamous rust fungi.
Really? As bad as rust?
Oh, yeah. A prime example is carnal bunt, Tillacea indica. It was first identified in India, but now it's a major global headache. When it showed up in Mexico back in the 70s, it triggered immediate quarantines on wheat shipments going into the U.S.
So what does it actually do to the wheat?
Well, the fungus essentially eats the inside of the wheat kernels, replacing the grain with masses of its own black teliospores. The infected kernels get partially or completely destroyed. And get this detail, they give off this really foul, rotting fish smell.
Oh, lovely.
Yeah, it's because of a chemical called trimethylamine. That's why they often call these stinking smuts. It tanks the grain's value, obviously makes it unfit to eat, and it can even cause allergic reactions in people working with the grain.
Wow. Okay, so they're not just ruining the crop yield, they're actually a potential fire hazard in storage and a health risk, too.
Oh, exactly.
But, okay, here's the twist. The really surprising upside, the aha moment. Those exact same unsightly galls from Ustalagomades on corn, the ones causing all that damage.
Yeah.
You can eat them. In Mexico, they're a culinary delicacy. It's called kutlacash, or sometimes maize mushrooms.
It is a complete paradigm shift, isn't it? Something seen as a disease, a blight, becomes this sought-after gourmet food.
It's amazing.
And in recent years, a commercial market for Quetzalcoatl has actually popped up in the United States. It's even led to research into how to artificially infect corn on purpose just to guarantee a good harvest of these galls for the food market.
So we're deliberately creating the disease now for food.
In a controlled way, yes. It's a fantastic example of, rethinking our relationship with organisms we previously just saw as pests.
That is incredible.
Yeah.
From enemy to, well, appetizer.
Yeah.
But their usefulness goes beyond the dinner plate, right?
Mhm.
You mentioned research.
Absolutely. Ustalagomates, the corn smut one, and another fungus called Microbacterium violaceum are incredibly important lab organisms. Geneticists, molecular biologists, they rely on these.
Why them specifically?
Well, take Microbacterium violaceum. It's often called the anther smut fungus. It does something truly fascinating to the plants it infects, usually plants in the carnation family. It sets up this permanent systemic infection, makes the host plant sterile for life.
Okay, that's pretty dramatic.
It gets weirder. It actually transforms female flowers into male ones. And then instead of the plant producing pollen, the fungus fills the anthers with its own spores.
Whoa.
And then insects come along thinking they're getting pollen, but instead they pick up and spread the fungal spores. The fungus basically hijacks the plant's reproductive system and tricks insects into being its delivery service.
That's some next level manipulation.
It really is. And studying how it does that gives us amazing insights into host pathogen interactions, gene regulation, things that can help us understand and maybe fight other diseases, even human ones.
Truly ingenious. Okay, so how does a fungus even operate inside a plant like that? Let's zoom in now. Go from that big picture impact down into the hidden world inside the plant.
Right. So in its infectious stage, the smut fungus exists in what we call the dikaryotic phase. This is the parasitic part, totally dependent on that living flowering plant.
And what does it look like inside?
It grows as these fungal threads called hyphae. They're usually slender, and they have these internal cross walls called septa. They often look quite convoluted, like tangled threads, and they typically grow between the host's cells.
Between them, not in them.
Usually between, yeah. Although some species, like Eustalago striformis, which infects Kentucky bluegrass, actually do grow through the host cells. But what's really striking is how the plant, or rather, how it doesn't react, for the most part.
What do you mean?
Most of the infected host cells show very little, if any, defensive response. They just keep growing, dividing, doing their thing, even with these fungal hyphae weaving around or through them. It's only right before the fungus is about to produce its spores that you start seeing tissue death, what we call necrosis.
So the fungus is like keeping the host alive, even growing while it spreads.
Pretty much. They're often called growth-altering parasites. They manipulate the host to their advantage. And if you look even closer at those septa, those cross walls in the hyphae, they often have this tiny central hole, a septal pore. It can actually close up rapidly if needed. Some species even have these flared walls around the pore called dollopore septa, though interestingly, they lack the cap-like structures you see with dollopores and some other related fungi.
Okay, so they're subtly manipulating the host, almost like keeping it functional but under their control. Now, you mentioned the dikaryotic phase being the infectious one. Is there another phase?
Yes, and it's remarkably different. It's called the homokaryotic phase. And unlike the eukaryon, this phase is totally non-pathogenic. You can grow it easily in the lab on simple nutrient stuff.
And what does it look like?
Often it grows just like yeast, as single individual cells. These single cells are produced by budding, kind of like yeast does, and they're usually called sporidia.
So one fungus, two totally different lifestyles,
Yeah?
a pathogenic thread-like one inside the plant, and a harmless yeast-like one you can grow in a dish.
Exactly. And here's where it gets really interesting, especially when we talk about mating. Most smut fungi are heterophthalic.
Hetero what?
Heterophthalic. It basically means they need two different compatible types to mate before they can become infectious. Those yeast-like sporidia we just mentioned. Compatible ones of different mating types have to find each other and fuse to form that pathogenic dikaryon. Think of it like needing two different puzzle pieces to click together.
Okay, like a fungal matchmaking system.
Precisely. And Ustalagomates, the corn smut, is the classic example we study to understand this. Compatibility is controlled by two separate genetic locations, or loci. They're simply called the A locus and the B locus.
A and B. Got it.
The locus basically controls whether they can physically fuse. It involves chemical signals, pheromones. If two sporidia have different versions of the A gene, say A1 and A2, they can recognize each other and their connecting tubes will fuse.
So A is like the handshake.
And an analogy, A gets them to connect.
Yeah.
But then the B locus comes into play. This one controls pathogenicity, the ability to cause disease. And unlike A, the B locus has many different versions, maybe up to 25 alleles.
25?
Yeah, lots of variations. So after the O loci allow fusion, the nuclei move into the fused cell. Now, if these two nuclei have different versions of the B gene, then you get the formation of a rapidly growing dikaryotic infection hypha. And that hypha causes the disease.
And if the B versions are the same?
If the B versions are identical, even if they fuse thanks to A, then nothing more happens. No infection hypha, no disease. It's this really elegant two-factor genetic control system.
That is a truly intricate system. Okay, so let's get back to the smut part, the actual black stuff. Those are the teleospores, right? The resting spores.
Exactly. They are the characteristic smut spores that give the group its name. They form in these dense masses called sori. And you can find sori in all sorts of host plant parts, flowers, seeds, leaves, stems, sometimes even roots. That's the visible, black, dusty stuff you see.
And how do they form?
It starts with a really dense growth of those fungal hyphae right at the spot where the spores will form. As the teleospores develop from these hyphae, the surrounding host cells just disintegrate. The mass of spores grows, and eventually it bursts through the plant's outer layer, the epidermis, exposing all those spores to the environment.
Is that when the galls form in corn smut?
Good question. With corn smut, Ustulagomates is actually a step before that. You get this massive overgrowth and enlargement of the host cells called hyperplasia and hypertrophy that forms the gall first, and then the fungus sporulates inside it.
Okay. And the spores themselves, how are they made?
They can form in different ways, sometimes singly, sometimes in chains, sometimes kind of budding off the hyphae. And their physical features are absolutely critical for telling species apart. It's like their fingerprint.
Right, you mentioned that. Size, shape,
color, yeah, yellowish, brownish, blackish. But the surface ornamentation is often key. Is it smooth? Does it have a net-like pattern, reticulate? Is it spiny? Got little bumps, tuberculate? You often need really good microscopes, like scanning electron microscopy, SCM, to see these fine details clearly.
And not always loose spores.
Correct. Sometimes they stick together in pairs or clumps. Or they can form these specialized structures called spore balls, which might have fertile spores on the inside and a layer of sterile cells on the outside, almost like a protective shell. Really diverse structures.
Okay, so these teleospores are formed, they burst out. What happens next? How tough are they?
Incredibly tough. They are resting spores designed for survival. They can sit in the soil for years just waiting, like little dormant time capsules.
And what wakes them up?
Their germination needs can be quite specific, even a bit weird. In temperate climates, low temperatures often actually favor germination. They might even need a period of chilling to break dormancy.
So they like the cold?
Often, yeah, in those regions, but maybe not in warmer areas. And other things matter too. Humidity, pH levels, even light can play a role, sometimes stimulating germination, sometimes suppressing it. It varies.
So they're tough, but also kind of picky about when they start growing again. What happens when one finally does germinate?
Okay, so inside the teleospore, before or during germination, a crucial event happens. The two nuclei fuse. That's karyogamy. This makes the nucleus diploid, having two sets of chromosomes. Then the spore germinates, typically by putting out a short hyphal tube called a promycelium, or sometimes metabasidium.
Promycelium.
The diploid nucleus moves into this promycelium and undergoes meiosis. That's the cell division that reduces the chromosome number back to haploid. This usually results in 4 haploid nuclei.
Right, by meiosis.
Then haploid basidiospores, which are often also called primary sporidia, form directly on the surface of this promycelium. And here's a really key characteristic of most smuts. These basidiospores are not forcibly discharged. They don't get shot off like in mushrooms.
They just sit there or fall off.
Pretty much. They just develop on the surface. And there's variation here too. The promycelium itself might be branched or not. It might develop crosswall septa or not. You can get few or many basidiospores forming on the sides or the end. And importantly, these basidiospores often start budding right away, like yeast, producing huge numbers of secondary sporidia or conidia.
Ah, so those secondary ones amplify the numbers.
Massively. And those secondary sporidia are usually highly infectious, although there's always an exception, right? One species, Tilletia fetida, a bunt fungus, can actually form spores that are forcibly discharged using a neat little water droplet mechanism called a Buller's droplet, but that's pretty rare for this group.
Okay, so mostly passive release, but massive numbers of secondary spores, and these are the main infectious agents.
Yes, generally the basidiospores and especially those numerous secondary sporia are what initiate new infections, though honestly studying exactly how they get into the plant can be tricky. They're tiny. They're transparent.
Hard to watch.
Exactly. And scientific reports sometimes even conflict. But we have some good examples. For carnal bunt of wheat, Tilicia indica, evidence points to the germ tubes growing out from the secondary sporidia and sneaking in through the plant's natural pores, the stomata.
Little breathing holes.
Right. For corn smut, Ustulagometes, we've seen sporidia landing on young leaves or the corn silks, finding a compatible mate, fusing via those conjugation tubes we talked about.
That A and B loci thing.
Forming that dikaryotic infection hypha, which then develops a special structure called an oppressorium, like a little pressure pad, and pushes its way directly through the host surface.
So direct penetration there.
Yeah.
But what about infecting the corn cob itself, the ovaries?
Ah, that's still a bit of a puzzle. For years, the assumption was it happened via the silks, but some newer research where they directly injected compatible sporidia into the developing cob got really high infection rates. So the exact natural pathway for ovary infection, well, it might still be a bit unclear. There's always more to figure out.
Fascinating. Always more questions.
Mm-hmm.
So this diversity in how they live, how they reproduce, it must make classifying them, figuring out their family tree, pretty challenging.
Oh, definitely. And that raises the big question. How do we categorize all these different smut fungi? Based on a lot of evidence, especially from their ultrastructure, the really fine details, and molecular data like RNA sequences, it looks like the main group, the order Ustilaginalis, is probably polyphyletic.
Polyphyletic, meaning they didn't all evolve from one single common ancestor for that group.
Exactly. It suggests that the smut lifestyle might have evolved independently multiple times from different ancestral fungi. Current thinking often points towards two major lineages. One group mainly parasitizes monocot plants, think grasses, like corn and wheat. The other group mainly hits dicot plants, broadleaf plants. And that dicot group seems potentially related to another fungal group called Exobasidium.
And there's other evidence for this split.
Yeah, subtle biochemical differences too. Things like the types of iron scavenging molecules they make, certain cell wall components, how they react to antibiotics. It all adds layers to the picture. And as a result, some species have actually been moved around. Like Ustilago violacea, the anther smut, is now in the genus Microbotrium. But the overall picture, the fungal family tree for smuts, is definitely still a work in progress.
Okay, so the big picture is complex and evolving.
Oh.
But historically, how are they grouped? Was there a simpler way people looked at them?
Yes, traditionally, smut fungi were often divided into two main families, the Astilaginaceae and the Tiliaceae, and this split was based primarily on how their teleospores germinate, specifically the structure of that promycelium.
Okay, so tell us about the Ustilaginaceae.
Right. In this family, the promycelium, that germ tube from the teleospores, typically kind of longish, lies flat against the surface, and it becomes transversely septate. It develops cross walls, dividing it into, usually, 4 cells.
Septate, got it.
And then the basidiospores bud off laterally from the sides and sometimes terminally from the end of this segmented promycelium. This family includes a lot of the really economically important ones we've mentioned, Ustilago avenae, oat loose smut, Ustilago tritici, wheat loose smut, and of course, Ustilagometes, corn smut.
And Eumetis is the research superstar too.
Yes, exactly. It's a fantastic model system because that haploid, yeast-like phase is easy and fast to grow in the lab. You can isolate all the products of meiosis to study genetics, and its mating system is relatively well understood. Super useful for research.
Okay, so that's Ustilaginaceae. What about the other traditional family, the Tiliaceae? How do they differ?
Their teleospore germination looks quite different. Typically, the promycelium is a septate, no crosswalls, and it's usually shorter, growing more upright away from the spore. And instead of spores budding off the sides, they typically produce a cluster of basidiospores, often about 8 of them, right at the tip of this promycelium, almost like a little crown.
A crown of spores. Nice image.
Yeah. And in a classic example like Tiliaceae caries, which causes bunt in wheat, meiosis actually happens inside the teleospore itself before it even germinates. That's a key difference. Then the haploid nuclei migrate into the promycelium, and the basidiospores form at the tip.
And their mating is different too.
Often, yes. In Talaciacaries, for instance, compatible basidiospores can fuse together via these little tubes, copulation tubes, while they're still attached to the promycelium. They form these distinctive H-shaped structures, and then these H pieces produce the infectious secondary sporidia, which are often crescent-shaped.
Wow, H pieces. Okay.
Mm-hmm.
Any other notable members in Talaciaceae?
Yeah, another one is Urocystis, which causes onion smut. It's known for making those distinctive spore balls we mentioned earlier, the ones with sterile cells on the outside protecting the fertile spores inside.
So this division based on germination, Astellaginaceae versus Talichiaceae, was it pretty solid?
Well, it was the traditional view, but it's not always perfectly clear-cut. The way a teleospore germinates can actually be influenced by environmental conditions.
So reality is messy.
Exactly. And researchers keep finding species with intermediate or variable characteristics. So while that two-family system is historically important, modern classification really tries to use a combination of features. Teleospore details, how the sorus develops, the symptoms on the host plant, which host plants it infects. It's a more holistic approach now, still complex, still being refined.
Right. Okay, so that really brings us full circle, doesn't it? We started with that unsettling image of black, sooty masses on plants. And we've journeyed through their roles as destructive pests, then as surprising edible delicacies, and even as vital tools for scientific discovery.
Yeah.
We've sort of peeled back the layers, looking at their internal structures, their really intricate ways of reproducing, those fascinating genetic controls for mating and infection.
Yep, the A and B loci.
And the ongoing challenges in figuring out their family tree, all starting from that one seemingly simple, soot-like spore.
It really shows the hidden complexity, doesn't it? And I think the key takeaway for you listening is how broadly relevant understanding these fungi is. It matters for global food security, definitely. These things directly impact crops that feed billions.
Can't overstate that.
No. And it's also vital for just fundamental biology research. Studying smuts gives us insights into genetics, how pathogens interact with hosts, evolution, just the amazing complexity of nature. And, despite everything we do know, there are still mysteries, those exact infection pathways, their deep evolutionary roots. Plenty left to discover.
Absolutely. It leaves you thinking, doesn't it?
Yeah.
Like next time you're maybe walking through a field or even just looking at, I don't know, cornmeal in your pantry, you might pause and think about this whole unseen world of fungi working constantly behind the scenes. And maybe wonder, how will our growing understanding of these microscopic battles continue to shape our agriculture, maybe our medicine, perhaps even what ends up on our dinner plates? And that wraps up our deep dive into the fascinating and, often surprising world of smut fungi.
It's been great digging into it.
Thank you so much for joining us on this exploration. We really hope you picked up a few aha moments along the way and feel a little more well-informed about these incredible organisms.
Hope so.
Keep learning, keep exploring, and we'll catch you on the next deep dive.